So good evening, and thank you all for being with us tonight, the fourth night of Brain Health Awareness Week. And if you missed our first 3 nights, you can still watch these by going to the University of Ottawa Brain and Mind website, where all of the links for each of the evening's events will be posted. So if you're just joining for the first time today, to introduce myself, I'm Ruth Slack, and I'm the director of the University of Ottawa Brain and Mind Research Institute. And before we get started, I'd like to introduce the Brain and Mind team who put all this week's events together. So I'd like to introduce Natasha Hollywood, our program manager.
Hi everyone, je vous souhaite la bienvenue.
Victoria Rache, our communications coordinator, and Candice Fortier, our administration coordinator. So on behalf of the entire team, I would like to extend a warm welcome to all of you tonight. And tonight's session is entitled Advancing Neuromuscular and ALS Research in Canada. And you may not be aware, but neuromuscular disease is a major strength at the University of Ottawa Brain and Mind Research Institute. And the University of Ottawa is home to the Éric Poulin Centre for Neuromuscular Disease. Researchers at this center are exceptional role models in translational research, which is bridging basic and clinical research. And they provide an inspirational training environment for trainees that come far and wide. So I'd like to begin the session tonight by introducing our first 2 speakers, Drs. Jody Warman and Dr. Robin Parks, who are the co-directors of the Éric Poulin Centre for Neuromuscular Disease. Dr. Warman is a clinician scientist in the Department of Medicine at the Ottawa Hospital. She's an assistant professor of neurology and holds a clinical research chair in the Faculty of Medicine. Dr. Robin Parks is a senior scientist and professor in the Department of Medicine. He is also the director of PhD research in the Department of Medicine at the Ottawa Hospital. And he also serves as the co-director of the Éric Poulin Centre for Neuromuscular Disease. So welcome, Drs. Parks and Warman.
Thank you for that kind introduction. So can you see my screen? Yeah.
Yep.
Perfect. Okay. So thank you everyone for coming. We're really excited to be able to talk about some of our really exciting research in neuromuscular disease and ALS in Ottawa. And we have no disclosures related to this talk. We've got a really exciting speakers tonight. Myself and Dr. Parks are going to be talking about the translational research in neuromuscular disease in Ottawa. Dr. Breiner, a fantastic clinician investigator, will be speaking about the ALS-related research and clinical trials. Dr. Gibbings will speak about his groundbreaking research in ALS research. Anjali Patel is a graduate student that will be talking about her motor neuron disease as a trainee, so a really unique perspective. And Dr. Locke Mueller is an international superstar in neuromuscular disease and clinician scientist, and he'll be talking about our new neuromuscular network in Canada. And so this night is slightly different than many of the other evenings in the fact that it's a neurological disease, but it's outside the spinal cord. So instead of Parkinson's disease or stroke or some of the other diseases that you might have heard of during this week, these diseases are really from the spinal cord all the way to the muscle. So motor neuron diseases such as ALS or spinal muscular atrophy, disorders of the nerve such as neuropathies like Charcot-Marie-Tooth or diabetic neuropathies, the junction disorders, which can either be autoimmune disorders such as myasthenia gravis or the congenital myasthenic syndromes, and finally diseases of the muscle such as the muscular dystrophies or some of the many autoimmune myopathies. And many of these diseases are very rare, affecting less than 1 to 2 or 5,000 people in the population, but there are over 150 different neuromuscular diseases, and collectively over 200,000 patients in Canada are affected with neuromuscular diseases. The University of Ottawa Centre for Neuromuscular Disease really joins the neuromuscular research and clinical care in Ottawa. So from the University of Ottawa, the Research Institute at the Ottawa Hospital, the Children's Hospital of Eastern Ontario, and the Heart Institute. And collectively, we have almost 70 clinicians and scientists doing fundamental research to clinical trials to gene-finding therapies. So we've got one of the highest concentrations of neuromuscular diseases in the world. And many of our researchers are world leaders for finding cures, for developing new therapies, for finding new genes. And we are very lucky to have this level of talent and research in Ottawa. And our goal as the Centre for Neuromuscular Disease is really to build a cutting-edge, world-class research program for basic research, for the translational research that Dr. Parks will talk about, and clinical research in Ottawa. We also want to lead a strong network of clinicians and scientists and develop really crucial patient links. And we also want to deliver a very collaborative research program for our superstar trainees for tomorrow. We need to make sure that we're building the next generation of clinicians and scientists. And one of our big projects over the past couple of years was to build a new neuromuscular research center. So CHEO is really well organized for the neuromuscular care, but we didn't have one center to take care of the large number of neuromuscular patients. So, there are over 10,000 patients in Ottawa that receive neuromuscular care. And so the new clinical research center was developed in 2018 and really has been a game changer for both the clinical care and the clinical research in Canada. And one of the things that we can do that's different is now we can do much more advanced laboratory investigations. We can provide comprehensive electrodiagnostic techniques and really push the non-invasive ultrasound and biopsies for patients and really move the research forward for the University of Ottawa researchers and the patients across the region. We can also provide really fantastic research opportunities for patients to participate in the natural history studies, which are important to study patients and figure out how diseases progress so that we can improve their care, exciting and innovative clinical trials to lead the best treatments, and as well provide, samples for our basic science researchers to help find the therapies for tomorrow. We have these really exciting translational research days. Well, we have disease-specific days, and this would be an example for having a basic scientist, nurse, technologist, clinician, administrative, and research coordinator. And we run these days in FSHD or spinal muscular atrophy or myotonic dystrophy. And this is really exciting for patients as far as bringing the best of clinical and translational research together. And we do this under a very comprehensive neuromuscular research ethics proposal that was developed through the University of Ottawa and the Ottawa Hospital and CHEO. And as my last point before I pass the microphone to Dr. Parks is I really want to highlight a very generous donation that's made a huge difference for neuromuscular care and research in Ottawa. And that is why we're called actually the Éric Poulin Centre for Neuromuscular Disease after Dr. Poulin. And Dr. Poulin was a real pioneer in minimally invasive surgery at the Ottawa Hospital from 2003 to 2013, and he was chief of surgery. And unfortunately, Dr. Poulin passed away of ALS, and his wife, in his honor, has given a very, very generous donation to the University of Ottawa Brain and Mind Research Institute and the Centre for Neuromuscular Disease. And this has really been able to change the clinical research and the basic and translational research in Ottawa. And this is Madame Brousseau when we were unveiling the plaque and tribute to Dr. Poulin. And so, her generous gift has provided a basic science postdoctoral fellowship in the Gibbings Lab, basic science and graduate scholarships that are helping our trainees move forward, clinical research and biobanking to provide samples to the basic scientists, which is fantastic, an endowed seminar series so we can bring even more exciting research and information in Ottawa, and finally a clinical research chair in ALS. And so I will pass the microphone to Dr. Parks to talk about linking the clinicians and the scientists together.
Thank you, Jody, for wonderful introduction. There's just a few points that I wanted to expand upon in my section here. And it's really the idea that truly impactful research requires collaborative research teams. So that the days of a scientist looking down a single microscope and coming up with world-renowned ideas is really over, and we need to collaborate with our researchers. We're so specialized these days that we do need to bring in different expertise to complement some of the work that we are doing. So whether it's a basic science researcher working with a clinical geneticist, but in reality, we need to get other people with other specialties like bioinformatics specialists, people who know how to manipulate data and understand it, physiatrists who give readouts for some of the therapies that we're doing, or diagnostic imaging specialists, and probably many others. And it's really these multidisciplinary research teams that are going to move research forward. So one of the things we're trying to do in Ottawa is break down the barriers between all of these different groups, and certainly starting locally. So the Center for Neuromuscular Disease has implemented a streamlined system to share samples and reagents between the basic and clinical researchers in Ottawa. And this is the bench to bedside and back research for NMD, where we can try to unite the basic scientists and the clinical researchers. And so the idea is that all patients attending the Ottawa Neuromuscular Clinic can contribute to the research in some way by providing DNA, blood, saliva, and tissue samples. So our clinical colleagues who you see when you attend the clinic, they can consent the patients and have the patients come in and provide these samples. And these are very precious samples because as a basic researcher, it's something that we don't have access to on a ready basis. And so these samples can be shared with the basic science researchers, where we can analyze those samples and try and understand the mechanisms of disease and apply some of those understandings to some of the other animal models that we have available to mimic these diseases, and hopefully come up with new approaches to treat these diseases that can then be passed into the clinic for eventual clinical trials. And that's really the way that we move research forward, is all working together. And really the idea is to try and translate this across Canada. So this isn't something unique to Ottawa. We want Canada to be, all researchers across Canada to be involved in this type of efforts. And this is largely come to fruition through the works of the Neuromuscular Disease for Canada Centre, which is headed by Dr. Locke Mueller, and I'm sure he's going to talk to you about this. But really this is uniting researchers across Canada. And why should we stop there? Really, it's a worldwide effort to come up with new treatments for some of the patients with disorders. So here in Ottawa, we actually signed a strategic partnership with University of Lyon. So Lyon has a very large neuromuscular research group, much like we have here in Ottawa. And the idea is to bring these two large centers together to work together to advance neuromuscular research. And these funds that we have available will support collaborative research grants, where one of the researchers is in Lyon, one of the researchers is in Ottawa, but also to allow for investigator and trainee mobility. And this will give the opportunity for our graduate students to study here in Ottawa and also in Lyon, and get the expertise from both sites. And we really are dedicated to trying to train the next generation of NMD leaders. We have a scholarship system that's in place. It's called the STAR Awards, the Scholarships in Translational Research. And these are funded through the uOBMRI and the kind gift from Dr. Éric Poulin. And actually, Anjali Patel, who is going to speak next, is one of the recipients of this award. So she'll talk a little bit about her research. We also can train clinical trainees here. So these are doctors that come in that are specializing in neuromuscular disease that can attend the clinics here in Ottawa and further their training in the understanding of neuromuscular disease and how best to treat it. Lastly, we also, of course, have our commitment to our patients, who are key to what we do. And we as a group are happy to go out and support any of the initiatives to either to meet with patient groups, go for the Walk for Muscular Dystrophy and try to raise some money for the families affected with these disorders, the Ice Bucket Challenge, and just any event to go out and talk about some of the research that's being done in here in Canada and the rest of the world, to update people as to some of the great work that's being done to improve our understanding, developing therapies for these disorders. So all of this together, really what we want to do is advance neuromuscular disease research, diagnostics, and patient care in Ottawa and of course across Canada and around the world. And the hope is to take today's research and turn it into tomorrow's therapies. With that, thank you. So what I would...
Attention, code white.
I'm very sorry about that.
North.
So what I'd like to do now is I would like to introduce our next speaker, who's Angelique Patel, who's a master's student with Dr. Max Rousseau, who's in the who's in the Department of Cellular and Molecular Medicine at the University of Ottawa. Anjali is a graduate student who received one of our STAR Awards, and the title of her talk is going to be Mapping ALS-Linked Protein Interactions. And I believe this is a pre-recorded message because as all good graduate students, I believe that she was busy studying and doing a test today.
Good evening, everyone. My name is Anjali Patel, and I'm a second-year master's student in the Rousseau Lab. In today's presentation, I'll be speaking on the objectives and techniques behind my project, which aims to study ALS at the cellular and biochemical levels. As you may have learned prior to this presentation, or will be learning soon, ALS is a progressive neurodegenerative disease that leads to the death of cells called motor neurons. Motor neurons are a special type of cell that transmits signals from the brain through the spinal cord and throughout the body to help control muscles and other organs. Their death results in symptoms that affect movement, including difficulties in walking, speaking, eating, swallowing, and eventually in breathing. ALS is a fatal disease and results in death approximately 2 to 5 years after diagnosis, and currently there is no effective cure. ALS affects about 1 in every 50,000 people, and there are currently about 3,000 individuals living with ALS in Canada. There is no single cause of this disease, and research has shown that it likely occurs due to interactions between genes and exposure to things in an individual's environment. Our DNA acts as a blueprint, telling our cells how to make different components which work together to perform different tasks or functions which are necessary for life. Mutations, which are changes in a person's DNA, can result in changes in cellular function and can lead to diseases like ALS. These components that are made by our cells using DNA as a blueprint are called proteins, and these proteins work together in different pathways and networks. Sometimes mutations in our DNA can give our cells the incorrect information, and this creates proteins that don't work like they're supposed to, resulting in dysfunctional pathways and leading to diseases like ALS. There are thousands of proteins found within our cell, each found in different compartments of the cell and playing a specific role there. Sometimes these proteins are transported to parts of the cell where they should not be, in a process known as protein mislocalization. And this is one of the overarching themes that we study in the Rousseau Lab. In ALS patients, we see protein mislocalization of a protein called TDP-43. So shown in these images here in brown, in cells from healthy individuals on top and ALS patients on the bottom, as well as depicted in red in our graphical cell models. Normally, TDP-43 is found in a part of the cell called the nucleus, which is also the part of the cell which contains our DNA. However, in ALS patients, we find TDP-43 mislocalizes and travels outside of the nucleus and forms these clumps called aggregates. These aggregates contribute to cell death. In our lab and in my project, we are interested in learning how this mislocalization of TDP-43 changes the way that the cell functions and what role this plays in ALS. And so to answer these questions, we will be using cells in combination with molecular biology techniques. Here I have a graphical representation of a single cell. As you recall, inside the nucleus of the cell is where the cell's DNA is contained. We can change the DNA inside the cell to model those of different individuals. For instance, we can have the DNA from healthy individuals, or we can add an ALS-causing mutation in order to model the DNA of ALS patients. On the slide here, I have a DNA mutation called the TDP43Q331K mutation, which is what I'm specifically studying through my project. By adding this mutation, we see TDP43 mislocalization from inside the nucleus where it is normally found to outside of the cell, or to outside in the cytoplasm. And this is also what we see in ALS patients. We are also able to add other DNA modifications to our cells, and as such, we inserted a gene called GFP that is normally found in jellyfish, which causes them to glow into our cells. So here on the bottom right, we have an image of some cells that are expressing GFP bound to TDP43, and we can see that the addition of GFP is causing our cells to fluoresce green. And so because of this modification, we are able to perform many different techniques on our cells. For example, we can look at our cells under a microscope and see how the Q331K mutation affects what part of the cell we find TDP43 in based on the fluorescent signal. In addition to this, and what I'm working on, is using these cells to study the network of proteins that TDP43 interacts and works with, both in healthy individuals and in the context of ALS. So in order to do this, I first start off by obtaining a dish of cells to collect samples of TDP43, as well as any proteins that are interacting with it. And I do this using a special type of bead, which acts as a magnet. Because we introduced the GFP gene into our cells, any TDP43 found in these cells is attached to the GFP protein, which these tiny beads are attracted to. And in this way, we are able to pull out TDP43 and its protein binding partners from our cells. These samples are then run through a machine called a mass spectrometer, which tells us the identity and the amount of each protein in our entire sample. We can then use this list of proteins in combination with data analyses programs, as well as literature searches in order to analyze pathways and to comb existing ALS patient data. And with this approach, we will then be able to narrow down and select an interesting candidate for further study. We will then be using a combination of cellular and molecular approaches to further study this protein. For example, we can knock out or remove this protein from cells entirely, or we can cause our cells to make large amounts of this protein and look at the resulting changes to cellular function in both of these cases. To us and to much of the field, these ALS-linked protein networks are complex and still largely unknown, and the pathways leading to neurodegeneration are not well understood. My project will allow us to shed light on proteins involved in these dysfunctional processes and to identify pathways that are involved in ALS. In addition to the Q331K mutation, we have the ability to make other patient-specific ALS-causing mutations in our cell lines, and then we can study the similarities and differences between all of these mutations. We can also expand our research to other disease models, including mice, flies, and patient-derived cells. These findings will not only allow us to increase our understanding of the disease itself, but will also equip us with the knowledge to design or identify new therapies or treatments that are uniquely designed for an ALS patient. And so I'd like to end off this talk by acknowledging the members of the Rousseau Lab, as well as my supervisor, my thesis advisory committee, and our collaborators for their support. As well, I'd like to acknowledge the OHRI Proteomics Corps for performing the mass spectrometry experiments, and to the Éric Poulin Centre for Neuromuscular Disease for funding this research. And so that concludes my talk for today. Thank you all for listening to my presentation.
So thank you, Anjali Patel, for a great talk. And just a reminder, if you have any questions, your questions can be submitted in the Q&A box, and we will answer these questions in the panel discussion at the end of the session. So now it's time to introduce the next speaker, Dr. Derek Gibbings, who is an associate professor in the Department of Cellular and Molecular Medicine at the University of Ottawa, and is also a member of the University of Ottawa Brain and Mind Research Institute. So welcome, Derek Gibbings.
Hi, thanks a lot. Just let me get my screen up. Can everybody see that, hopefully? So I'm associate professor in the University of Ottawa and a researcher in the Éric Poulin Centre for Neuromuscular Disease. And today I'd like to talk to you about work we do in our lab that aims to understand the causes of ALS and to develop new drugs to treat patients. So as Anjali just told you very nicely, amyotrophic lateral sclerosis is a progressive neurodegenerative disease in which motor neurons die. And the motor neurons basically allow signals in the brain to communicate with the muscles. So when those motor neurons die, then the body loses its ability to control key muscles controlling movement, breathing, and swallowing. And this leads to patients dying usually within 3 to 5 years of getting this disease. So over the last 30 years, with large advances in genome sequencing technologies that you can see on the right side of this graph, we've identified a lot of different kinds of genes in labs across the world whose mutation causes ALS. But that leaves still about 80% of ALS cases with an unexplained cause that might be a mix of gene and environment, as Anjali mentioned. So one of our key questions in the lab is what causes motor neurons to die in ALS? And it's a difficult question because we have these observations in patients that we can see at pathology, but we can't really do experiments in patients. So we have to come up with model systems, either in cells that we can grow in dishes or in animal models that resemble that disease as closely as we can. So one of our key things we know about ALS is that in 95% of patients, again, as Anjali introduced very well, we see these aggregates or clumps of a protein called TDP-43. And so those clumps of this protein are thought to be one of the major causes of ALS. And so what we try to do in the lab is to understand what causes these clumps to form and ways that we might eliminate them to help to understand and treat the disease. So one of the things that's come out of the literature in the past decade is that a lot of these proteins which are mutated in ALS all localize in these things called stress granules in cells. And so in a normal cell, these proteins tend to be diffusely distributed throughout the cell. And then when cells encounter stresses, these things all aggregate into these little dots or clumps of proteins that you can see at the microscope. And so the thing that's quite striking is that if you look across a lot of the genes whose mutation causes ALS, the proteins made by those genes, especially when they're mutated, drive the formation of these kind of clumps of proteins in cells called stress granules. And so the model that's emerging in the literature is that these mutations in ALS prevent motor neurons from adapting to stress, and that might cause them to die. And so this is actually just a short video showing kind of what that looks like if we look at it by video under a microscope. You can see that in a normal cell, the proteins are in the nucleus, and then they rapidly form these granules, and then those things can slowly disappear as the stress goes away. So where the literature has come to is a model in which if you have formation of too many of these stress granules or clumps of proteins, then you are likely to get ALS. Where our lab has made a significant contribution is in actually understanding a pathway that eliminates those stress granules. So over the past couple of years, we've published some papers that have shown that this unique cellular waste removal pathway called autophagy can basically recognize these stress granules, swallow them up, and degrade them. And so what we think our research has helped show in the field is that if you get too many stress granules in cells, it's likely to lead to ALS. And that can happen by making too many of the stress granules with some mutations, or by preventing the elimination of those stress granules by autophagy. And so our first goal in the lab is to basically challenge this model in a way. So we have this list of 30 genes that we know cause ALS, but in fact, our knowledge about ALS really has only studied a couple of those genes in any detail. And so we want to look at all of those 30 genes and how they control basically the formation and the elimination of these stress granules. And the goal there is threefold. One is it challenges this model. Is it true or false that these stress granules are really critical to the disease? And by doing this, we can identify new mechanisms that control their formation, and that can lead us to new ways to treat the disease. So a second goal we're interested in the lab is to engineer new drugs to prevent the formation of these clumps in patients with ALS. So we currently have 2 drugs approved for ALS, but they really have minor benefits for patients. So there's a new class of drugs that's in late-stage clinical trials, mostly from a company called Ionis. And these drugs show great promise to really drastically slow down disease. So they work by basically stopping the production of mutant proteins. And they can do that by 50 or 80%. So you only have about 20% of that disease-causing protein left. The problem is that they only work in some inherited forms of ALS, so a small percentage of patients. And they also require up to a 12-hour injection into the spinal cord, and they have some risks of toxicity. So we wondered if we could maybe come up with something that might be a little bit better for patients. And in our minds, making smart drugs often comes from using solutions that nature has evolved. And so in our approach, we're using nature's delivery service or postal service. So our bodies make, all the cells in our bodies make these tiny bubbles called exosomes that naturally flow around our body and our blood and carry materials between cells. So what we did is we engineered these tiny bubbles called exosomes to carry gene silencing drugs, like I just mentioned to you, and made them so that they could get into the brain and the spinal cord. So we can inject these into the blood instead of the spinal cord. We can similarly reduce expression of the mutant genes that cause ALS. And with our approach, it's more efficient. It requires 100 times less drug, so it's safer. And it's an all-natural product that the body is naturally in the body, and so it's safer. And so we're currently testing those in mouse models of ALS and Alzheimer's. And some of the first results here on the right show that we're increasing the ability of these ALS disease mice to move and the strength of their muscles. And so we're actually having an effect on these disease. And so we're pushing forward with this approach, trying to build safety and toxicity information and generate manufacturing processes to produce it in a way that we could use it in patients. A second approach that we're using in the lab, again, uses another kind of naturally inspired approach, and that's nature's waste removal service, this autophagy process that we know can basically recognize, swallow up, and degrade these clumps of proteins that cause ALS. And so we engineered a way to basically direct autophagy to specifically recognize and degrade the TDP-43 aggregates that cause ALS. And again, comparing in a cell model to kind of the next best drug, with our approach, we're eliminating a lot of those aggregates compared to the kind of normal disease type cells. And so we think that we're very excited about this approach, because with this approach, you could actually treat the 95% of patients that have these TDP-43 aggregates. And as well, most of the time, by the time patients are diagnosed, they've accumulated these protein clumps for decades. And so the gene silencing approaches can actually get rid of that stuff once it's actually there, while this approach can actually recognize and eliminate those clumps already. So I'll end there, and I just want to say thank you to all the people that work in the lab. So this is the people who work on ALS-related research in the lab, students and other researchers. Funding is essential to keep the research process moving through the Éric Poulin Centre for Neuromuscular Disease and some other sources. And research collaborators, again, as was mentioned, people in Ottawa, in Toronto, at MIT, have all made this research happen. Thank you very much.
Thank you, Dr. Gibbings. That was a great talk. And we'll save our questions for the end, but I would encourage everyone to put their questions and type them in because we'll take them at the end of the talk. And I would like to introduce Dr. Ari Breiner. Dr. Breiner is an assistant professor and clinician investigator at the Ottawa Hospital, and he really has led and spearheaded the new clinical trials starting at the Neuromuscular Centre at the Civic campus. And I had a patient tell me once that he's not only an excellent neurologist, but a really fine gentleman, and I would agree with that. So I'll turn the screen over to Dr. Breiner.
Okay, thanks very much for the opportunity. So it's my pleasure to shift gears a little bit and to talk about an update on clinical research in ALS in terms of medications that are in use and active clinical trials of medication. So these are my disclosures. So as you've heard, ALS or Lou Gehrig's disease is a debilitating and eventually fatal neurodegenerative disease that causes progressive paralysis of the muscles. And the disease burst onto the public consciousness in 1939 with the diagnosis of Lou Gehrig, who was a professional baseball player for the New York Yankees. When his diagnosis became public, he made an amazing statement, and he referred to himself as the luckiest man on the face of the earth, despite this devastating diagnosis. And it's really that sort of resilience and positive attitude that we see in our ALS patients, and really which inspires many of us to work on the clinical care and research in ALS. I'd like to shift now and talk about ALS treatments. So before 1995, there were really no effective treatments available for ALS. But in the mid-1990s, there were 2 clinical trials published which showed a survival benefit of 3 to 5 months for a drug known as Riluzole or Rilutek. And that's a drug that blocks the toxic effects of glutamate on motor nerves. The drug is relatively simple to use. It's an oral tablet and has minimal side effects. But I think the big drawback, as Dr. Gibbings and others had mentioned, is that the survival benefit of 3 to 5 months is relatively mild. Unfortunately, between 1995 and 2017, there were a number of failed clinical trials in ALS research. It's kind of a dark area in ALS research. And there's a number of reasons why trials failed in that interval, including a number of drugs that were tested with poor penetration of the brain or spinal cord, some drugs that had weak preclinical data, and in other cases, imperfect outcome measurement in clinical trials. But recently, I guess since 2017, we've seen some progress in ALS research, first as a result of a medication known as edaravone. So edaravone is a free radical scavenger for peroxyl radicals and peroxynitrite, and has protective effects, not just on nerve cells, neurons, but also on glial cells, support cells against oxidative stress. When tested in mouse models of ALS, it was shown both to delay the progression of the clinical disease of the symptoms, but also to delay the disease pathology on a cellular level. And as a result, it was tested in a large phase 3 clinical trial, which recruited ALS patients who had early disease and intact breathing function. And this phase 3 study showed a 1/3 or a 33% reduction in the rate of decline of function. And the results were published in a very high impact medical journal, the Lancet Neurology. What this publication in 2017 meant was that it was the first time in 20 years we had an additional agent that could actually slow the progress of ALS and provide a number of additional months of survival benefit on top of Riluzole alone. So it was a very exciting development in the field of ALS. Very shortly after the publication of this study in the Lancet Journal, the medication was approved by the US Food and Drug Administration. This was in the fall of 2017. But unfortunately, the drug was not immediately available to Canadian ALS patients. And as a result, patients in Canada began to seek alternative means of access to the drug, either by traveling to Japan to have it administered and to purchase the medication, or by making arrangements for the medication to be shipped from Japan to Canada, and therefore they were able to have it administered closer to home. Thankfully, by October 2018, the medication did receive expedited approval from Health Canada, but despite this approval, there were still challenges that faced patients in accessing the drug. This was now as a result of funding constraints and some disagreements between the manufacturer and payers, insurance payers and government payers, about the appropriate price for the medication. And as a result of these logistical hurdles facing patients, a few of us in the ALS community wrote an editorial to the Canadian Medical Association Journal describing the challenges facing Canadian ALS patients trying to access edaravone. And we compared this to a lifeboat ethics situation where equally deserving individuals were fighting to access lifeboats on a sinking ship. Thankfully, within the past 1 to 2 months, I'm pleased to say that these access issues seem to have been resolved, and there seems to be equitable distribution of edaravone across Canada for appropriate ALS patients. I'd like to shift now to some very new or hot off the press data, which was published last month in September 2020 in the New England Journal of Medicine regarding a new medication known as AMX0035. It's a combination drug of 2 medications known as sodium phenylbutyrate and taurursodiol or TUDCA for patients with ALS. What this phase 2 study showed was a benefit in terms of slowing the progression of the disease. And these medications were targeted to a new or novel mechanism looking at reducing stress at the level of the endoplasmic reticulum, as well as reducing mitochondrial dysfunction. And again, this phase 2 study showed a slower decline in the ALS functional rating score for patients who were on active drug as compared to the group that received placebo. And perhaps more exciting was the results of an open label extension study of this medication, the sodium phenylbutyrate TUDCA, where in the open label extension, all patients received active drug between months 6 and 36. And what they showed was that at the 3-year point, patients who had received active drug from the beginning seemed to show a survival benefit of an additional 7 months as compared to those who had received placebo at the start. And I think this data, the survival data combined with the slowing data, shows that this medication warrants a more definitive and large phase 3 study. I'd also like to highlight an interesting and very recent paper, which has not yet been published, but is in pre-publication state, looking at observational real-world evidence surrounding the use of riluzole. As we know, the initial clinical trials in the 1990s seemed to show a very modest 3 to 5 months survival benefit with riluzole. However, this second look actually highlighted a number of real-world observational studies showing median survival of 6 to 19 months longer in patients who had taken riluzole as compared to those who had not. And so what this highlights is that perhaps riluzole is actually more effective than we think it is and than we are quoting to patients. I do get many questions from patients surrounding stem cell treatment in ALS. And I would note that much of the excitement has surrounded the phase 3 study conducted by Brainstorm for their product known as Neurons, which are spinal injections of mesenchymal stem cells. Unfortunately, Brainstorm did not select any Canadian sites for their phase 3 study, and they elected to conduct their study at 6 sites in the United States. So Canadian patients had very limited access to participate in this study. But I think it's very exciting to know that this study has completed recruitment, and we're expecting some data for presentation and publication within the next 6 months. So I think what I would say is stay tuned to determine whether stem cells are indeed an exciting development in the world of ALS. And then finally, before wrapping up, I want to mention that 1 or 2 successes with edaravone and AMX0035 and potentially stem cells, we don't rest there. One success really only gives us more enthusiasm to test other new drugs in the pipeline. And I think it's a very rich time right now for ALS drug development. There are quite a few drugs in the pipeline. And I always encourage patients who are diagnosed with ALS to participate in clinical trials, either here in Ottawa at the Éric Poulin Centre, or at other Canadian neuromuscular centers of excellence. And there are a variety of studies that are recruiting patients here and elsewhere. So with that, I will wrap up. I want to give thanks to the University of Ottawa, both the Brain and Mind Research Institute, as well as the Éric Poulin Centre for Neuromuscular Disease for supporting clinical as well as basic science research in ALS. I want to thank colleagues in the neuromuscular and ALS clinics, as well as, of course, patients with ALS and other neuromuscular diseases.
Well, thank you, Dr. Breiner, for a great talk, very inspiring. So it's now a pleasure to introduce our last speaker, Dr. Locke Mueller. And I think I can speak for all of us, but we're all very excited just over 2 years ago when Dr. Locke Mueller was recruited to the University of Ottawa. Dr. Locke Mueller is a neurologist and a clinical academic specializing in genetic neuromuscular disorders and rare diseases. He trained as a neurologist in Munich and at the Montreal Neurological Institute. He held a faculty position at the Institute of Genetic Medicine in Newcastle in the UK before coming to the University of Ottawa. He's now a senior scientist at Geo Research Institute and a professor of neurology in the Faculty of Medicine, University of Ottawa. And he's involved in many international networks and now leads the Neuromuscular Disease Network for Canada. So thank you, Dr. Locke Mueller, for being with us today.
So thank you so much, Dr. Slack, to introduce me. I'm delighted to be here tonight and give a further talk after many exciting and highly interesting talks. I wish to sort of broaden this out a bit further. So we heard a lot for very good reasons about ALS, and ALS is obviously one of the most important neuromuscular diseases. But there's many more neuromuscular diseases out there. And in fact, many more rare diseases. And of course, many face similar problems like ALS. And I want to sort of highlight those themes that have already been mentioned earlier, but maybe in a slightly different context. So as Dr. Slack just kindly said, I was recruited to Ottawa just 2 years ago with help of the Brain and Mind Research Institute. And my focus is on rare inherited neuromuscular disorders. And there's a variety of them, such as myotonic dystrophy, muscular dystrophies, and congenital myasthenic syndromes. And my approach is translational, meaning we do bench to bedside with basic research, animal and cell models, genomics research, and then also bringing this back to the clinic with clinical trials such as Dr. Breiner just explained. And our emphasis is on Canada, Canadian patients, but with international collaborations. And as was said before, collaborative work is essential for progress in rare disease. And we want to support Ottawa's position on the global map for rare disease research. So why do rare diseases matter at all? Because of course, rare disease often is felt to be maybe less important than common disease. But rare diseases do actually affect a lot of people because each of these rare diseases and rare neuromuscular diseases are individually rare, but there's 9,000 different diseases out there, so they make a large number of people. But because each individual rare disease is rare, often even your family doctor would not know what that disease is, or patients say, my doctor told me I cannot do anything for you. I don't know where to start. And that has a lot of knock-on effects. So there is even sometimes a lack of an exact genetic diagnosis. As we heard from Dr. Warman, there might be a lack of appropriate data and samples for research, lack of cohorts for trials, resulting in a lack of interest from the pharmaceutical industry to develop such treatments. And that all is, of course, a hindrance to make progress in rare disease. But as I just said before, and was mentioned before, it's actually a large number of people that are affected by rare disease. So we estimate about 8% of the population, 3 million people in Canada, and more often these conditions are lifelong conditions, chronic, and therefore make up quite a large part of the healthcare spending. But there's another part, another side of the coin, because these rare diseases are often genetic. We do often understand at least where these conditions start. So they have a clear gene or mutation that's underlying them. And therefore, we can start unraveling the pathways that lead to these neuromuscular diseases through modern technology like genomics techniques. And that also gives us a handle to develop targeted genetic and advanced therapies. There is an international research consortium that was launched pretty much 10 years ago, and that includes international funders like the NIH institutes, but also the European Commission, or in Canada, CIHR and Genome Canada. And they set out that, in fact, no, not even a country or a big country like the US alone would be able to find better cures and treatments for all these different rare diseases. So they need to pull together as a consortium, and this is the International Rare Disease Research Consortium. And I, as well as Dr. Boycott from the GEO Research Institute, were both involved early on as chairs of scientific committees, and we set out in 2010 with pretty ambitious plans to develop 200 new therapies for rare disease and to find means to diagnose most rare disease. And these objectives were set to be reached within 10 years by pulling these funders together and large amounts of money dedicated to this goal. In fact, this was so successful that 200 new therapies were achieved in 2016, so 4 years ahead of schedule. And we are still hunting for more disease genes because it turned out maybe that there's more rare diseases out there than we originally anticipated. However, even now, there is still a huge unmet need for rare diseases, both for research and for healthcare, because if we continue to develop 200 new therapies every 6 years, maybe, then it will still take another 200 years until each rare disease has at least one therapy. And that would be assuming that this is always like one therapy per one rare disease, which is probably not the fact. So what helps us to sort of accelerate this development? Well, targeted treatments will rely on better diagnosis and next generation sequencing. So looking at all genes in the human body and in a patient really has helped to find a lot more genetic causes for rare disease and make them in a way treatable. And I'll switch gears to show you one example, just how this collaboration works for one individual rare disease. A very talented student in my lab, who's now a postdoc at GRI, Dr. Emily McConnor, found some years ago that there was a new gene, a myosin gene, or called myo9A, that causes a childhood onset neuromuscular disease that we call congenital myasthenic syndrome. And these kids have difficulties with swallowing. They have weakness of most muscles. They even have problems with breathing. They have a characteristic ptosis, which is the hanging of the eyelids that you see in the images. And they do respond to some conventional drugs to some extent. But we found this new gene and explained the condition in these kids by looking at actually two different families, one that had two affected SIBs and another one that has just one affected child. So what turned out was that in both of these families, it was mutations that inactivate the myo9A gene so that there wasn't enough myo9A, and we suspected that myo9A would be the cause of the myasthenic syndrome by having an important function for muscle and the connection between nerve and muscle. But that was a speculation at the time, so we set out to actually find proof for this hypothesis. So first of all, we used neurons, so nerve cells in culture, and removed the myo9A gene. And what we found in these cells is that they changed their shape and their cytoskeleton formed unusual fibers that we didn't see in the normal cells. And we felt that maybe there's a change in the proteins in these cells. And similar to the talk that you heard earlier, we did a mass spec experiment where we looked at all the proteins in these cells. And we didn't stop there. We also looked at all the proteins that were secreted from these cells to see whether there's a difference in those proteins between cells that have the myo9A gene and cells that have the myo9A gene inactivated or depleted. And what we found was that actually there was quite a lot of proteins that were either upregulated or downregulated secondary to the myo9A defect. The ones that are in the cell that were down or up are labeled in green and red. And they were actually the minority, while the majority of proteins that showed changes were actually those proteins that were secreted out of the cells. So these are the proteins that are labeled in blue and in brown. Quite a large number of proteins, but one particular protein caught our attention, which is written here. It's the so-called Agrigene gene. And the reason for that is that we already knew that Agrigene plays an important role in the neuromuscular junction, helps actually form and stabilize the neuromuscular junctions. So we speculated that based on the defect in the myo-9A gene, the nerve cells secrete less agrin, and that destabilizes neuromuscular junctions and leads to the muscle weakness. So in order to look at that in vivo, so in an animal model, we used so-called zebrafish embryos. So they develop very fast after fertilization. Within 48 hours, they become zebrafish embryos that actually swim independently in the tank. So they have a fully formed neuromuscular system. And we can easily, through injections into these fertilized eggs, inactivate genes like the myo-9A gene and then see whether the fish are still able to swim and still have a functioning neuromuscular transmission. So what we see here in these traces, actually zebrafish embryos are very small. You can almost not see them with the naked eye. But what we see here in blue is the trace, it's the distance a zebrafish embryo that has the myo-9A gene inactivated can swim. So that is actually for a zebrafish embryo a fairly short distance. However, if we add that molecule to the zebrafish that we felt was reduced secondarily in a lower concentration, 0.5 microliter of agrin, then the trace looked like that, and the zebrafish could swim about double the distance as when there was no agrin given. And if you give even more agrin, double the dose, then the red trace comes, which is a sort of further improvement of the swimming ability of the zebrafish. So basically what we show here is that through investigation in these models from cells to zebrafish, we were able to figure out what one of the likely reasons were why this genetic defect leads to a neuromuscular condition. And secondly, how a potential therapeutic approach would look like. And similar approaches are now actually tested, going into tests with patients in clinical trials. I switch gears a little bit and talk about the neuromuscular network. So as you have seen, because of the rarity of these conditions, to have sufficient numbers of patients to go into clinical trials, this is not possible to do in a single center, even as large as Ottawa. So we need to pull together resources. And these resources are, of course, patient cohorts, but they are also samples from biobank, they are data, they are experts, etc. And therefore, the CHR and Muscular Dystrophy Canada have teamed up to provide funding for 3 years in the first instance to set up a neuromuscular disease network for Canada that we call NMD4C. That was launched in January and has been taken up enthusiastically. So we now have 284 members from Vancouver to Halifax across most provinces of clinicians, researchers, but also patients, families, and advocates to help this translational research and networking agenda. We have a lot of information on our website, and I invite you to look it up. And we are particularly proud of our early career researchers that contribute to the network and will be the next generation. We set up specific initiatives for them, such as a curriculum, so to make sure that they all sort of highly trained both for care, but also for research and neuromuscular research. The steering committee that is in charge of the neuromuscular network is actually heavily biased towards Ottawa and the Brain and Mind Research Institute with Dr. Warman as well as Dr. Kotari. But we are also having Dr. Dowling from Sick Kids in Toronto, and Stacey Lintern from Muscular Dystrophy Canada. And there's many more people who very actively contribute, and I invite all of you to not only sign up to our newsletter, but also bring your ideas, join us as members, and become part of the network. So I end here, and I would thank you all for your attention, and of course many collaborators across Canada and in various countries, as well as of course the Brain and Mind Research Institute and the Éric Poulin Centre. Thank you.
So thank you, Dr. Locke Mueller, for a very exciting talk and sharing some of the research that's happening in your lab. And also I'd like to thank all the other speakers for great talks this evening. And now we have the opportunity for a panel discussion where you can submit your questions again to the Q&A box. And Natasha Hollywood, who is our program manager, will lead the question period.
Thank you very much, Dr. Slack. I'll ask maybe all the panelists to maybe open up the cameras in preparation for the Q&A. Great, thank you. Okay, our first question is, this is for the panel. Are certain groups of people more susceptible to neuromuscular disease? In other words, is NMD strongly dependent on genetic inheritance?
Maybe I can start. So there are over 150 neuromuscular diseases. So there is a disproportionate number of genetic diseases. I think it depends which region that you're from. There are certain diseases that are more prevalent. For example, in Canada, we've got one of the highest rates of myotonic dystrophy in the world, or oculopharyngeal muscular dystrophy. But there are a number of diseases that are autoimmune and not genetic. And there are certainly people that can have both. So it's a simple question and a complicated question at the same time. But there are many, many types of genetic neuromuscular disease subtypes that all have different genetic mutations.
Dr. Warman. This is also for the panel. The normal prognosis after the first diagnosis of ALS is only 2 to 5 years. And yet Dr. Hawken, the celebrated theoretical physicist who, even after early onset of ALS, lived a fair long life full of extraordinary and original contributions. How can that be possible? Are there different types of ALS, or was it simply the exceptional genetic makeup of Dr. Hawken?
So I think there's a few points and questions within the question. The fact that Dr. Hawken made exceptional contributions is very typical. Many of our ALS patients are extremely bright and individuals who contribute to society, write, publish, are very active individuals, and despite the physical limitations are still extremely intellectually active and excellent advocates. In terms of Stephen Hawking, his story is certainly remarkable. He was diagnosed with ALS in his 20s, and he passed away in his early 70s of the disease. So certainly that's a much longer trajectory than we see with most patients. However, for every rule, there is an exception. And I will say that even in our local clinic, we see patients who live longer than the textbook definition of ALS. So we certainly see individuals who live more than 10 years with ALS. And perhaps the young onset of the disease in Stephen Hawking in part was protective. Certainly we do notice that younger patients may have longer survival with the disease. But for every rule, there's an exception to the rule, and I think it's not unreasonable that he lived a long time with ALS. Thanks.
Thank you. This is for Dr. Gibbings. What would be the challenges to using exorcisms to treat patients?
Yeah, so there are several challenges. I mean, it's a complex biological entity. So a lot of drugs that we have traditionally are kind of, you know, a relatively simple chemical structure, whereas this is much larger and delicate. And so it takes a lot more care to produce it. It's a lot more work to produce large amounts of it. And so that's one challenge that probably would make it a more expensive drug, like a lot of other drugs that are emerging these days. I mean, one of the other challenges would also be that that there could be some safety issues around people having immune responses, because it would almost be like having a little transplant from one patient to another. So we traditionally would have thought that you might have had an immune response to these exosomes when you gave them to somebody else, but now people have actually done that in a number of scenarios in animals and even patients, and that doesn't seem to be the case. But there's always unknowns in these processes. So there's never, you know, with any drug development, as I mean, Dr. Breiner mentioned, there's been so many drugs that have been tried, and a lot of them have failed for a lot of different reasons. There's never any kind of clear home shot run for any of these efforts, but you know, we all try our best and try to get them there. You're on mute.
Thank you. This is for the panel. My grandmother died of ALS in the 80s, and my sister has MS. Is there a link? Do I need to be concerned? Should I go for a DNA or some sort of testing? I am 65 years old, and so far I'm healthy and I'm not on any medication. Thank you.
I don't.
Sure. Yes. Oh,
Ari.
that's okay. You know, we in a forum, it's always hard to comment on any specific case, and you know, it's not really a forum to give out advice or information on any particular case. But I will say that in general, we don't feel there's an overlap between ALS and multiple sclerosis. Multiple sclerosis is believed to be an autoimmune condition of the central nervous system that results in loss of myelin, which is the protective covering of the nerves within the central nervous system, not thought to be linked with ALS. The diseases where we do see overlap with ALS is some of the dementia conditions, particularly frontotemporal dementia. And we also see overlap between ALS and Parkinsonism, or signs of Parkinson's disease, in some patients, particularly with very specific genetic mutations. And so really, it's the overlap between ALS and other neurodegenerative diseases, not overlap between ALS and other and autoimmune diseases. So my answer would be, I don't think the two are linked.
Great, thank you. Okay, so the next one would be, how do you participate in the Neuromuscular Disease Registry? My brother and mother have MS, and I have, and I apologize for the pronunciation for this, the dermatomyositis. Sorry about that. No one ever asked for genetic material. Whoever answers can please maybe say that word properly for the person asking the question.
Maybe I can jump in here. So as Dr. Breiner has just explained, multiple sclerosis, or MS, would not be a neuromuscular disease, so would also not then be captured in a neuromuscular registry. But I'm sure there's similar types of data collections in the MS field, and maybe at one of the other evenings of the Brain and Mind Institute that's being covered. So in terms of the dermatomyositis, so that's an inflammatory muscle disease, or also an autoimmune disease. There's certainly activities in terms of clinical trials. It's shared across neurology and rheumatology, but we are certainly very interested in dermatomyositis, and if the patient would see us at the Neuromuscular Centre, then we could talk about things like samples and registry, etc.
Unmute. Okay, so the next question would be, the focus is very much on mutated genes as a cause in the presentation of some. Dr. Gibbons states that 80% of ALS patients seem to be caused by unknown causes. How much time and funding actually does the brain mind spend the possible environment causes factors that might lead to the stress granules as mentioned in the two presentations?
I can maybe start for a first bit, and then Dr. Gibbings can add if he feels he wants to add anything. So I think for many diseases, even if they're autoimmune or not caused by genetic factors, understanding the genetic underpinnings of certain diseases can help many. And I'll give the examples of statins. So many people are on Lipitor or Crestor or one of the statin medications for cholesterol. And there's over 200 million people worldwide that are currently taking statins. And statins were developed after a few families with very, very high cholesterol called hypercholesterolemia. And these treatments were developed for these families, but they were found for patients with non-genetic causes. And this has saved millions of lives for patients that have heart attacks and strokes. So finding the underpinnings of diseases, even if they're not genetic, or how the genes interact is really critical to moving many therapies forward. Now, that being said, there are many researchers at the University of Ottawa Brain and Mind Research Institute that are looking at non-genetic causes and the interactions between different proteins that also look for the autoimmune, inflammatory, environmental causes. I don't know if Dr. Gibbings or Dr. Breiner want to add to that.
Sure, I'll jump in. I mean, I think one of the other issues is that, a lot of the things we found about is one single gene causes the disease, but sometimes it's kind of mutations in 5 different genes that might cause the disease. And that's a lot harder to track down experimentally, would be one thing. So there could be genetic causes, but they're just harder to understand and we haven't understood them yet. And I think the other real challenge is we'd all love to understand this 80%, and there's been a lot of work out there to try to track down environmental causes, but it's really hard, and you end up with basically epidemiological data where, these people all, had this lifestyle, did these activities, were in these environments. And statistically, with a small number of ALS patients, I think it's just really hard to track down confidently an environmental factor that we could say causes or one of these diseases. So, there's a few factors that have a minor increase in risk for ALS. And then I guess the last thing I would say is that as well, kind of in the lab when we're trying to study these things, we're often studying mouse models, and it's hard to really know and convince ourselves that we're really looking at something that's ALS if we're using a chemical or something to induce it. So we're always concerned that we might not be studying the right thing. Whereas if we take the genetic mutations that cause the disease, I think we're just a bit more sure that we're really studying a mechanism that causes what we see in patients. And so we have a bit more confidence in that approach. So it's just a really complicated area to study the environment.
I would agree with everything Dr. Gibbings said. I think that in a rare disease, studying exposures is nearly impossible. To start tracking, water supplies and chemical exposures. And, as Dr. Gibbings mentioned, there are some rare things that have been weakly associated with ALS epidemiologically, but the evidence really isn't strong. So when we study the disease, we take the minority of patients that have a known genetic mutation that have exactly the same clinical presentation. So their symptoms are exactly the same, sporadic disease and genetic disease. And we say in the sporadic disease, we have no idea what's triggering this cascade of motor neuron loss, but in the genetic form we do. So let's go at attacking the causes where we know it and try to learn lessons from that we can then apply to the 90% of sporadic cases.
Thank you. So I'll just ask two more questions. The next one would be for Dr. Breiner. What does participation in a clinical trial entail for ALS patients?
Participation in a clinical trial, I would say it's not for everyone. Participation in a clinical trial obviously is having access to a cutting edge or new medication, but may also have unknown side effects. As well, anyone participating in a clinical trial has to be aware that there's a placebo arm, so they may be receiving active drug or may be receiving placebo. And generally, it's a commitment to follow through with using the study medication, being self-aware and tracking side effects, and attending follow-ups, usually on a monthly basis at our Neuromuscular Centre in order to carefully track strength and breathing function and other outcomes that are important.
Great. And the last one would be, does brain injury increase the risk of ALS?
Maybe Dr. Gibbings has something to say about this as well, but there are some weak links. So there are some pathology studies showing a link between repeated head trauma, like in boxers and professional athletes, and ALS-like pathology degeneration within brain tissue on autopsy studies. Yeah, so there are some links, but I think there's more to be elucidated there.
Yeah, I don't think I have too much more to add. I mean, I think there's, you know, in terms of environment, there's, I think, a group of Italian soccer players who, but nobody understands why they had a higher rate of ALS. It could be, you know, some people might say chemicals in the field, some people that might say head injuries from repeated heading of the ball. I think military veterans in the United States have about a twofold or so higher risk of ALS. Again, you know, there's lots of factors that could be. One of the hypotheses is brain injury and concussion, but again, it's really hard to know what's just a correlation and what's a causation, right? I mean, you know, maybe it's all because, you know, they ate too much Kraft dinner, or, you know, whatever or, you know, they all went to the same pool, or who knows, right? I mean, it could be anything. We would never know. Or they all had the same genetics and came from the same background, you know, or something.
Well, thank you very much. I'll pass, thank you much for the panel discussion on the Q&A. I'll pass the mic over to Dr. Slack. Thank you very much, everyone.
All right, so I'd like to again thank all the speakers for their exciting presentations. And also I'd like to thank our partner institutions. And today we're highlighting the work from the Ottawa Hospital and the Children's Hospital of Eastern Ontario. And of course, I'd also like to thank the Brain and Mind team. And if you would like to support our research at the University of Ottawa Brain and Mind Research Institute, please go to our website and go to the link, The Power for Change is Yours. And also, if you'd like to get more information and attend our future events, go to the Brain and Mind website. Those will all be posted, and follow us on Facebook and on Twitter. And again, thank you so much for being with us tonight.